The mucosal barrier is the primary physiological interface between an animal's internal milieu and the external pathogen load. It is an active boundary, demanding localized, specialized adaptive responses to maintain homeostasis. Historically, studies of avian mucosal immunity focused almost exclusively on the surface areas of the respiratory, gastrointestinal, and reproductive tracts. Recent data requires an expansion of that model. The avian skin, and specifically the feather pulp, operates as a localized, critical component of the mucosal immune system. During experiments with Marek's disease virus, researchers documented a significant influx of T cells directly into the feather pulp between 4 and 10 days post-infection. These images display longitudinal sections of the feather's proximal ends. Dense brown staining confirms distinct accumulation of CD4 and CD8 positive cells following vaccination. This infiltration is accompanied by localized upregulation of cytokine genes, indicating complex immune responses occurred directly within the feather pulp. While the feather pulp presents a new immunological frontier, the gastrointestinal tract remains the largest and most structurally complex mucosal compartment in the bird. To understand avian gut immunity, we must first recognize a distinct anatomical constraint. Birds lack the highly structured, encapsulating lymph nodes that define mammalian immune responses. The avian system compensates for this absence through an extensive network of gut-associated lymphoid tissue, or GALT, dispersed directly along the digestive tract. This network possesses a unique phenotypic signature, maintaining high concentrations of TCR gamma delta positive T cells, also known as TCR1 cells. Furthermore, the intraepithelial lymphocytes in the gut show a high prevalence of CD8 alpha-alpha homodimers, contrasting with the CD8 alpha-beta heterodimers typically found in systemic circulation. Avian enteric competence relies on the precise coordination of this spatial architecture, specialized non-mammalian T-cell subsets, and a time-dependent posthatch developmental cascade. This structural integration allows the avian gut to balance microbiome tolerance with the rapid clearance of diverse pathogens. The primary architectural and functional unit of the avian small intestine is the crypt-villus axis. This histological section traces the axis from the stem cell zone in the crypts of Lieberkühn up to the apical tips. Enterocytes migrate along the villus structure over 48 to 96 hours before shedding at the tip. Beyond their role as a physical barrier, these specialized enterocytes execute active secretory functions. This animation illustrates the secretion of Muc2 mucins, creating a physical barrier segregating commensal bacteria. At the crypt base, the avian intestine completely lacks mammalian Paneth cells. Instead, the mucosal surface secretes avian beta-defensins, which disrupt invading pathogen membranes. The enterocyte layer functions as a highly active innate sensory and defense organ. Moving deeper past the epithelial barrier, we reach the underlying connective tissue, the lamina propria. This map details specialized avian GALT structures distributed along the digestive tract, including the cecal tonsils, Meckel's diverticulum, and Peyer's patches. The overlying epithelium contains specialized M cells that sample luminal contents, delivering antigens directly to underlying dendritic cells. The cellular makeup of the intraepithelial lymphocyte compartment, or IEL, differs entirely from the profile seen in systemic blood and spleen tissues. The IEL is virtually devoid of B cells. It is dominated instead by CD8 positive T cells and the specialized TCR gamma delta positive population. The lamina propria presents a contrasting profile. It is heavily populated by B cells that have already undergone class switching. These lamina propria B cells execute high-volume production and directional transport of secretory polymeric IgA directly into the gut lumen. This spatial separation, a T-cell dominant epithelium above an IgA-producing lamina propria, compensates for the lack of lymph nodes, hardwiring defense directly into the mucosal architecture. At hatch, the neonatal chick faces a severe developmental constraint. Its gut is functionally naive, poorly developed, and virtually devoid of structured lymphocyte cellularity. Immune competence is acquired through 3 distinct T-cell export waves from the thymus. The first wave occurs pre-hatch, seeding the periphery. A pronounced second wave brings a surge of T cells right after hatch. Finally, a third reinforcing wave expands the populations. The environmental commensal microbiome serves as the catalyst. The physical complexity and clonal expansion of the T cell repertoire relies on these microbial colonization triggers. Functional capacity maps directly to these cellular waves. The ability to mount effective oral tolerance or mature antibody responses is absent until these waves conclude. Avian immune competence is not a baseline state. It is a microbially triggered structural buildout that dictates survival during the first weeks of life. We can observe the consequences of this developmental timeline by looking at Salmonella enterica, serovars enteritidis, and typhimurium as a test case for luminal bacterial clearance. The clinical outcome correlates directly to the thymic export timeline, separating neonates under 3 days old from chicks older than 3 weeks. In a 3-day-old chick, the naive, highly permeable gut allows unrestricted systemic translocation of the bacteria into deep tissues, resulting in fatal disease. By 3 weeks of age, the mature GALT architecture restricts Salmonella to the lumen. The system utilizes rapid heterophil infiltration and flagellin TLR5 interactions to contain the bacteria. This introduces a physiological paradox. Salmonella infection induces significant proliferation of B cells and exceptionally high titers of antigen-specific IgA. However, experimental data proves that chicks rendered permanently B cell deficient via surgical bursectomy clear primary and secondary enteric salmonella at the exact same rate as wild-type birds. This demonstrates that B cells and secretory IgA are not required to clear this luminal pathogen. The host relies instead on redundant non-B cell mechanisms. The avian gut challenges classical mammalian assumptions. Significant secretory IgA production does not necessarily equate to the primary mechanism of protective immunity. The ultimate contrast to luminal salmonella is Eimeria, an intracellular protozoan parasite responsible for coccidiosis. Eimeria presents a distinct biological challenge. Its extremely short intracellular replication cycle demands rapid, highly specific immune intervention to prevent tissue necrosis. The effector requirement for clearing Eimeria is strict. Unlike Salmonella, clearance is completely dependent on TCR alpha-beta 1 CD4 positive T cells. The actual mechanism of parasite attrition is mediated by the targeted production of interferon gamma by these specific T cells. Depleting other gut subsets, like the CD8 positive or TCR gamma delta positive T cells, only delays clearance. But depleting the TCR alpha-beta 1 population entirely prevents immunity. This highly targeted T cell response places intense genetic selection pressure on the parasite, allowing researchers to map specific protective antigens like AMA1 and IMP1 for subunit vaccines. The avian enteric immune system achieves defense without classical lymph nodes by perfectly integrating its spatial architecture, synchronized cellular ontogeny, and customized pathogen-specific effector deployment.